Hello dear students, welcome back to Biovidyalaya. I hope you all have well prepared for our CSIR NET examination that will be held on 17th September. As you know that Unit 6 System Physiology Plant is very important topic for your CSIR NET examination. You can score minimum 20 marks from this unit. So today we are going to discuss CSIR NET Plant Physiology questions from December 2018. So let's start. First question, which one of the following mineral deficiency will first be visible in younger leaves? Options Option 1 calcium, option 2 nitrogen, option 3 zinc, option 4 molybdenum. So from these following options we have to find which mineral deficiency first we can observe in younger leaves. Okay, you have to remember that initial symptoms of nutrient deficiency we can visible either in new leaves, that means younger leaves, or in older leaves. Uh, Minerals such as nitrogen, phosphorus, potassium, manganese deficiency are first we can observe in older leaves, while some immobile nutrients are there. For example, copper, manganese, calcium, sulfur, nickel. Such mineral deficiency first we can observe in younger leaves, that means in new leaves. Okay, so here answer is calcium, because this calcium is needed for plants to produce new growing points and root tip. So deficiency of this calcium will lead to younger leaves to curl downward with browning of leaf edges and leaf tips. That is known as tip burn. That will lead, if calcium is deficient in that plant, that will lead to tip burn. Okay, here is the explanation. Answer is option A, calcium, because calcium is needed by plants to produce new growing points and root tip. In general, initial symptoms of nutrient deficiency is expressed either in new or older leaves. For immobile nutrients in plants, for example, nickel, calcium and sulfur, the deficiency symptom first shown up in the younger leaves. Okay, so answer is option A, calcium. The carbon dioxide compensation point for C3 plants is greater than C4 plants because in C3 plants, option 1 dark respiration is higher, option 2 dark respiration is lower, option 3 photorespiration is present, option 4 photorespiration is absent. So from this following option you have to find why in C3 plants carbon dioxide compensation point is higher as compared to C4 plants. Okay, we know that Calvin cycle means fixation of carbon dioxide by the enzyme ribosco. Okay, and finally sugar will synthesis, energy will produce. But this ribosco can fix both carbon dioxide and oxygen. When atmospheric oxygen is more as compared to carbon dioxide, then ribosco will fix oxygen instead of carbon dioxide. Then it will lead to the loss of fixed carbon dioxide, that will release carbon dioxide. So this will lead to loss of energy. Okay, but in C4 plants, ribosco is present in bundle sheath cell instead of mesophyll cells. In mesophyll cells, phosphoenol pyruvate carboxylates, that means PEP carboxylase is present, that can fix carbon dioxide. They are not this PEP carboxylase is not able to fix oxygen. So this PEP carboxylase will fix carbon dioxide, then oxaloacetic acid will form, then oxaloacetic acid is converted to malic acid, then it will transport to bundle sheath cell. In bundle sheath cell, ribosco is present. Okay, so in bundle sheath cell, this malic acid will release carbon dioxide, then ribosco will fix this carbon dioxide, and then it will enter into Calvin cycle. Okay, so due to the higher concentration of carbon dioxide in the bundle sheath cell, photorespiration is minimized. Okay, And in C3 plant, photorespiration is higher, but in C4 plants, it is negligible or absent. Okay. So, carbon dioxide compensation point in C4 plants is low, but it is greater in C3 plants. So, answer is option 3, photorespiration is present. In our one of the lecture video, we already discussed about C3 cycle and C4 cycle. If you didn't watch that video, please do watch. So this is the explanation of that answer. Carbon dioxide compensation point, carbon dioxide fixed in photosynthesis is equal to carbon dioxide evolved in respiration and photorespiration. The value is 25 to 100 ppm in C3 plants and 0 to 10 ppm in C4 plants. The reason for low compensation value for C4 plant is greater efficiency of carbon dioxide fixation through PEP carboxylates. C3 plants photorespiration is high and leads to loss of fixed carbon dioxide. Okay. Next question. Which one of the following best describe the function of Casperian bands during the translocation of nutrients and water across the root? Option 1. Block apoplastic nutrient transport. Option 2. Block symplastic nutrient transport. Option 3. Act as a nutrient carrier. Option 4. Help in creating passage cells. So, from this following option, we have to find the correct option that is describing the function of Casperian bands during the translocation of nutrients and water across the root. So, first what will happen? First, root will absorb water, then this water will move through different cells and finally it will reach xylem. Okay. So, which are the different cells? Epidermis, then cortex, then endodermis and pericycle and finally xylem. Okay. And there are two pathways, Apoplast pathway and Symplast pathway. In Apoplast pathway, water and nutrient will move through cell wall and intracellular space. And in Symplast pathway, water will move from protoplasm of one cell to another via plasmodesmata. Okay. We already discussed about the difference between Apoplast and Symplast pathway. If you didn't watch that video, then I will give the link in the below description. Please go through that. Okay. So this Casperian strip or Casperian band is present in the cell wall of endodermis, and this is a lignin-like structure or wax-like substance that is deposited as a ring in the transverse section of endodermal cells, and that will block the flow of water and nutrient through this apoplast area. That means through this cell wall area. Okay, so it will block apoplast pathway. Okay, so option 1 is the correct answer, block apoplastic nutrient transport. Okay, so please try to watch that video, the difference between apoplast and symplast. So here is the explanation. The Casperian strips are lignin-like structure that is deposited as a ring in the transverse section of endodermal cells and block the passive flow of water and solute in the apoplast. Okay. So correct answer is option 1, block apoplast nutrient transport. Okay. This is very easy question. Which one of the following components is expected to be most abundant in the phloem sap of a plant? Option 1 protein, option 2 organic acid, option 3 sugars and option 4 phosphates. We all know that phloem is for the transportation of sugar and xylem for the transportation of water. And the sucrose is the main sugar in the phloem sap. So answer is option 3 sugar. Okay. So this is the explanation. Sugars and amino acids are most predominant metabolites in the phloem sap. Sucrose is the main sugar in phloem sap and its concentration varies between species. So answer is sugar. This is part C 4-mark question. Following observations were recorded while studying physiological parameters of sorghum and wheat under similar conditions. Statement A. Sorghum ribisco exhibits relatively higher affinity for carbon dioxide compared to that of wheat. Light saturation. Next statement B. Light saturation of net photosynthetic flux is relatively lower for sorghum compared to that of wheat. Statement C. Warburg effect is difficult to record for sorghum and could be said as not measurable, whereas it could be easily recorded for wheat. Statement D. Temperature optimum for net photosynthesis is lower for sorghum compared to that of wheat. Statement D. 13C by 12C ratio of assimilate is relatively higher for sorghum compared to that of wheat. Which one of the following combination of the above observation is correct? So these are the options. Okay. So from the above statements A, B, C, D and E, you have to find which are the correct statement. For answering this question, you must know which is C3 plant and which is C4 plant. This sorghum is C4 and wheat is C3 plant. Okay. First statement A. Sorghum ribisco exhibit relatively higher affinity for carbon dioxide compared to that of wheat. This is not correct. Okay, because C3 ribisco has high affinity for carbon dioxide in order to minimize reaction with oxygen. You all know that C4 plants ribisco is present in bundle sheath cell because its affinity for carbon dioxide is less. That's why it is present in bundle sheath cell instead of mesophyll cells. Okay. So sorghum ribisco exhibit relatively lower affinity for carbon dioxide. So here the statement is not correct. Statement B. Light saturation of net photosynthetic flux is relatively lower for sorghum compared to that of wheat. So what is meant by light saturation point? Point where the light intensity does not increase the photosynthesis rate is called the light saturation point. In C3 plant, it is 50 to 70 percentage. It is 50 to 70 percentage in C3 plants. Okay, but it is 20 percentage in C4 plants. So it is lower for C4 plant. That means sorghum. So this statement is correct. Light saturation of net photosynthetic flux is relatively lower for sorghum. That means it is lower for C4 plants compared to that of wheat. That is correct. Next, statement C. Warburg effect is difficult to record for sorghum and could be said as not measurable, whereas it could be easily recorded for wheat. So, what is meant by Warburg effect? The rate of photosynthesis decrease with increase of concentration of oxygen, and this phenomena is known as Warburg effect. Okay, it is easily measurable in C3 plants, but it is difficult to measure in C4 plant because there is a spatial separation in initial carbon dioxide fixation and Calvin cycle. Initial carbon dioxide fixation is occurring in mesophyll cell and Calvin cycle is occurring in bundle sheath cell. So it is difficult to measure in C4 plant. So this statement is also correct. Next statement D. Temperature optimum for net photosynthesis is lower for sorghum compared to that of wheat. This is not correct. C3 plant photosynthesis decrease as temperature increase, and optimum temperature for net photosynthesis is between 25 degrees Celsius to 35 degrees Celsius in C3 plant. But in the case of C4 plants, photosynthesis rate remains constant with increasing the temperature. And its optimum temperature is between 30 degree to 40 degree. Okay, so this is higher for C4 plant as compared to C3 plant. So here it is given that lower for C4 plant. Lower for sorghum means lower for C4 plant. So this is not correct. Next statement E. 13C by 12C. This is isotypes of carbon. 13C by 12C ratio of assimilate is relatively higher for sorghum. That means it is higher for C4 plants compared to that of wheat. This is also correct. This ratio is higher for C4 plants. Okay, this is correct. So which are the correct statements? B, C, E. See here option 2. Only B, C and E. This is the correct answer. If you are getting such questions in your examination, then try to find out the incorrect answers. Then you can easily answer this question. Okay. So this is the explanation. The incorrect statements are A and D. C3 plants containing ribosco that has higher affinity for carbon dioxide than C4 plant, which help minimize oxygenase reaction. C3 plants photosynthesis decrease as temperature increase. The optimum temperature for net photosynthesis lies between 25 degrees Celsius to 35 degrees Celsius. In C4 plant, photosynthesis remains roughly constant with increasing temperature, and optimum temperature for net photosynthesis lies between 30 degrees Celsius to 40 degrees Celsius, which is higher than most of C3 plants. Okay. This is also part C 4-mark question. Following are certain statements regarding respiratory metabolism in plants. Option A, respiratory quotient during partial breakdown of carbohydrate. That means alcoholic fermentation will be infinity. Statement B, respiratory quotient indirectly provides information about nature of substrate used for respiration, the relative rate of competing respiratory process. Statement C, breakdown of organic acid in mature fruit will exhibit a respiratory quotient value of more than 1 since organic acids are relatively oxygen rich compared to the other common substrate. Statement D, anabolic metabolism can influence respiratory quotient by removing reduction equivalents leading to decrease in oxygen uptake. Which one of the following combination of the above statement is correct? So these are the options. Okay. First we can discuss some points related to RQ value. Then we can discuss the question. So what is meant by RQ or respiratory quotient? The ratio of volume of carbon dioxide released to the volume of oxygen taken in respiration is called respiratory quotient, and it is denoted by RQ. Okay, so RQ is equal to volume of carbon dioxide released divided by volume of oxygen taken. Okay, and this knowledge of respiratory quotient help in determining respiratory substrate. It help in knowing the type of respiration being performed. It provides some information about major transformation of food materials. Okay, so these are the significance of respiratory quotient. When carbohydrate is respiratory substrate and it is completely oxidized, then RQ is always unit. That means RQ will be 1. In this process, the volume of carbon dioxide released is always equal to volume of oxygen used. So, if carbohydrate is completely oxidized, then RQ is equal to 1. Okay. In some succulent plants like Opuntia, Bryophyllum, etc., carbohydrates are incompletely oxidized to organic acid in dark without the evolution of carbon dioxide. Hence, RQ remains 0. Okay. In anaerobic respiration, carbon dioxide released but oxygen is not used. Hence, in this type of respiration, value of RQ is infinity. This is important. In anaerobic respiration, RQ will be infinity. Okay. For example, alcohol fermentation. Okay. When substances rich in oxygen are respired, the value of RQ is more than unity, more than 1. For example, organic acid. Okay. When substances rich in oxygen like organic acid are respired, RQ will be more than 1. Okay. An RQ value of fat and proteins is less than 1. RQ value of fat and proteins is less than 1. And for carbohydrate, it is 1 if it is completely oxidized. Okay. And lesser is the RQ value, more is the energy released. Okay. Lesser is the RQ value, more is the energy released. So RQ value means respiratory quotient, that is the ratio of volume of carbon dioxide released to the volume of oxygen taken. Okay. So here we have to find the correct statement. Okay. So based on that information, we can find the correct statement. Okay. Try to find out at least 2 correct statement, then we can easily answer this question. Okay. So let's see how to solve this question. Before that, you have to remember that molecules that are more oxidized like glucose require less oxygen to be fully metabolized and therefore they have higher respiratory quotient. But in the case of molecules that are less oxidized like fatty acids that require more oxygen for their complete metabolism and they have lower respiratory quotient. Okay, just remember that. Okay. First statement A, respiratory quotient during partial breakdown of carbohydrate, that means alcoholic fermentation, will be infinity. Okay. So, alcoholic fermentation means anaerobic respiration. So, in anaerobic respiration, respiratory quotient will be infinity. This is correct. Okay. So, let's see the option. Here, only A is there and in fourth option also A is there. But in second option and third option, statement A is not given, so we can eliminate second and third option. Okay. So let's see which option, option 1 or 4, which is correct. Next statement B. Okay. Respiratory quotient indirectly provide information about nature of substrate used for respiration, the relative rate of competing respiratory process. This is also correct. If you are not sure, then just go for the next statement. Okay. Statement C. Breakdown of organic acid in mature fruit will exhibit a respiratory quotient value of more than 1 since organic acids are relatively oxygen rich compared to other common substrates. You have to remember that in the case of oxygen rich substrate, RQ value will be greater than 1. Okay. That means value will be more than 1. So this statement is also correct. So we can confirm that option 4, A, B, C and D. This is the correct answer. So statement A, B, C and D are the correct statements. Okay. So option option 4 is the correct answer. Seventh question. See elements of phloem contain sugars and other organic materials throughout the plant. The following statements were made about characteristics of C elements in seed plants. Statement A. Angiosperms contain C plate pores. Statement B. There are no C plate in gymnosperms. Statement C. P protein is present in all eudicote and many monocots. Statement D. There is no P protein in angiosperm. Which one of the following combination is correct? Option 1 B, C and D. Option 2 A, B and C. Option 3 A, B and D. Option 4 A, C and D. Here we are not doing the detailed explanation of this question because we already discussed this question in one of our lecture video called P protein. In that video we did the detailed explanation of this question. So please go through that video. The link is here in the below description. Okay. So here we have to find the correct statements. Okay. So here some statement about C elements are given. First statement is angiosperm contains C plate pores. This is correct. In angiosperm, C plate is present. Okay. And there are no C plate in gymnosperm. This is also correct. In gymnosperm, instead of C plate, C areas are present. So this is also correct statement. Then next C. P protein present in all udicot and many monocots. This is also correct. P protein is present in dicot and monocot. So this statement C is also correct. Last statement D. There is no P protein in angiosperm. This is not correct because P protein is present in angiosperm. Okay. So this statement is incorrect. So which one of the following combination is correct? A, B and C. So let's see the option. Option 2, A, B and C. Okay. So option 2 is the correct answer. This is the last question. The plant hormones auxin and cytokinins and their interaction play an important role in regulating apical dominance. The following figure represents an experiment related to study of gene interaction that influence axillary bud outgrowth or dominancy. Q, Z and M represent genes involved in phytohormone pathway. So here in this picture we can see that X, Z, M like that. Okay, this Q, Z, M represent genes involved in phytohormone pathway. Okay, based on the above figure, the following statements were made. Statement A, X is an auxin that maintain expression of Q and Z and repress M. Statement B, Y is a cytokinin that promote axillary bud growth and is induced by M. Statement C, decapitation, that means removal of apex, activate X. Statement D, X is a cytokinin that repress M. Which one of the following options represent correct statement? Option 1 A and C only, option 2 B and D only, option 3 A and B only, and option 4 C only. So this question is based on apical dominance, and based on this picture we have to find which of these following statements are correct. Okay. So here Q, Z and M represent genes involved in phytohormone pathway. Okay. If you know the experiment that is related to apical dominance, then you can easily answer this question. Okay. So what is meant by apical dominance? Apical dominance means growing shoot apex that will inhibit the growth of axillary bud. Okay. Growing shoot apex that will inhibit the growth of axillary bud. That is called apical dominance. If you remove the shoot apex, then axillary bud will grow. That means lateral bud will grow. Okay. So this is intact plant with shoot apex. Okay. So here you can see that the hormone X, that is moving from the tip to base, and that time the gene Q is on. Here gene Q is off. In axillary bud, this is axillary bud. In axillary bud, gene Q is off. And here Z is on and M is off. And here you can see that after decapitation, that means after the removal of shoot apex. Okay. The hormone X is absent here, and there is one hormone called Y that is moving to the auxiliary bud, and here Z is off and M is on. Okay, here Z is on, but here Z is off. Here M is off, but here M is on. Okay. So after decapitation, there is no hormone X. Okay. But here hormone Y is there, that is moving to the auxiliary bud. And here here auxiliary bud outgrowth. So here you can see that after auxiliary bud outgrowth, hormone X is coming to this auxiliary bud to the base of the plant. So we can say that this hormone X is moving from tip to the base. That means that is transported basipetally downward from tip to the base. Okay. At that time Q is on, Z is on, and M is off. Okay. So we can say that X is auxin because it is moving basipetally. Okay. After decapitation, there is no hormone X. So we can confirm that X is auxin. And here Y is there. This Y is leads to the growth of auxiliary bud. So we can say that Y is cytokinin. So let's see the statement and we can confirm that. X is an auxin that maintains expression of Q and Z and repress M. Yes, this is correct. Here you can see that X is present. So Q is on and Z is also on. But M is off. So it is repressing M. Okay. And it will list the expression of Q and Z. So this statement A is correct. Statement A is correct. Next, statement B. Y is a cytokinin that promotes auxiliary bud growth and is induced by M. See, here Y is there, that is leading to the growth of auxiliary bud. Okay. So, here M is on. Okay. So, it is inducing M. So, this statement B is also correct. Next, C. Decapitation, that means removal of apex activate X. No. Here we are not seeing X, hormone X after decapitation. So this is not correct. Next, D. X is cytokine that repress M. No. X is auxin. Okay. So this is also not correct. So which one of the following options represent correct statements? Which are the correct statement? A, B. So here option 3, A and B only. So option 3 is the correct answer. Okay. So this is the explanation. I hope this video was helpful for you. If it is helpful, please share this video with your friends. So here I am winding up this lecture video. If you like this video and find it helpful, then please click the like button, share this video with your friends, and also don't forget to subscribe this channel for getting more videos like this. Keep learning. Thank you.